Am I audible?
Yes, it's audible.
Yeah. I'm starting the presentation now.
Okay.
Yeah, good afternoon, everyone. I'm Sranjeev Rangaraj. I'm a PhD scholar from the Department of Biochemistry, University of Hyderabad. And also, I'm a Prime Minister Research Fellow. To complete my associate, I have associated with this NPTF course called Overview and Integration of Cellular Metabolism. And we are in the week 5. So in the last 4 weeks that we have majorly seen about the carbohydrate metabolism, digestion, and all those things, then how the carbohydrate metabolism plays major role in the cellular energy and then the function. Last week we have started with lipid metabolism, lipid digestion, and then the absorption that we have seen it in the last week. And as usual, we'll see recap on the week 5 concepts. Then we'll do some practice questions, and if you have any doubt, we'll do the doubt clearing session. Uh, Majorly, how the transportation of the lipid occurs. That's what the topic of concern this week, and we'll see about how this lipids are getting metabolized inside the cell. For example, catabolism of the lipid or free fatty acids in terms of the beta oxidation. That's what we are going to see overall in this uh, 1 hour. We know that majorly lipids are hydrophobic molecule, and due to the presence of carbon and then hydrogen molecule, and these are not soluble in the aqueous medium of plasma. But whereas if you see all the enzymes that are related to lipid metabolism, those things are work very well in the aqueous media. So there should be some carrier molecule or the transport material that should make this lipid molecule, which is hydrophobic to hydrophilic. Okay, so that's what we are going to see. To transport these molecules or this lipid molecule, it should be converted into the water soluble. That will be done by, or that these lipid molecules will be solubilized by the converting them into the lipoprotein. And after converting into the lipoproteins, what will happen? It will have the both hydrophobic as well as the hydrophilic group. Then it can be freely transported into plasma as well as in the lymph tissues. And if you see overall, the definition of lipoproteins are conjugated lipids. It will have two major things. One, it can contain the lipids as well as the protein molecules, as the name suggests. The lipids also, it can be an hydrophobic lipid or the amphiphilic lipid. Amphiphilic are nothing but it has both positive and then the negative charge. That is called amphiphilic lipids. And in the hydrophobic lipids, we have triglycerides, cholesterol esters, and phospholipids, and then the cholesterol. And if you see, cholesterol esters are more hydrophobic than cholesterol. And finally, the protein component of lipoproteins can be varied from ApoAs, ApoBs, ApoCs, ApoE, D, and then Apo small a. And other proteins also will play major role in this lipoproteins constituent. And this is the overall structure of lipoprotein. If you see, it has, as I already suggested, it can have the polar surface where it formed by the glycerophospholipids and then the cholesterol. And there will be the non-polar lipids that is inside that will be formed by the cholesterol esters and then the triacyl glycerol. So this is the main overall structure of the plasma lipoproteins. As the name suggests, it should be hydrophilic. Only then it can help in the transport of lipid molecule. So overall, as I already suggested, this lipoprotein functions as a transport vehicles, and this for transportation of the insoluble form of the lipids into the blood plasma. Either it can be the exogenous lipid particles or the endogenous lipid particle. Exogenous in the sense the day-to-day dietary sources, whatever that we are taking. from lipids or from oil items, whatever that we take, any kind of non-veg, these kinds also will have the cholesterols. Then from one organ to the another also will be done by this lipoproteins. So we are going to see what are the different types of lipoproteins present in our body, and how it is helping in the transport of the lipids. As we all know, there are inverse correlation between the diameter of the this lipoprotein as well as the density of the molecule. So the chylomicron is the lowest density having, it's a very lowest density lipoprotein in the overall uh, lipoprotein classification, and the source of this thing is majorly it's in the intestine. And if you see the density, it's less than 0.95 gram per ml, whereas the next one is the chylomicron remanence, and the source is from the chylomicron itself, it will be formed, and this density is little bit larger than the previous one. And VLDL are majorly present in the liver as well as in the intestine. So it is otherwise called as very low density lipoprotein. Then the IDL, and it is formed from the VLDL, and that is a little bit higher density than the VLDL. Then finally the LDL, that is low density lipoprotein, that also having 1.019 to 1.063. And finally HDL. HDL is the high density lipoproteins, which is formed by liver, intestine, and then VLDL, and then the chylomicrons. And it has very different varieties of classes: HDL1, HDL2, and HDL3. The precursor molecule for the HDL is P beta HDL, that is having high density in the system. And there are other lipoproteins such as the albumin and then the free fatty acids. This we will see it before beta oxidation, how albumin, those things are playing an important role in the system. And this forms in the adipose tissue, and that is having the highest density. If you correlate with the diameters, the chylomicron has the highest diameter when compared with the pre-beta HDL. And if you see the main lipid components in the these different lipoproteins, in chylomicrons, the 98 to 99 percentage of the contents are lipid molecule. For example, here we talk about the triacyl glycerol. The content of triacyl glycerol in chylomicron is so high, whereas the protein content like this apolipoprotein A2, A3, A5, B48, these things are 1 to 2% only in the chylomicron. The next thing is chylomicron remanence, which has the triacyl glycerol, phospholipids, and then cholesterol. It has 92 to 94 percentage of the lipid content, the same 6 to 8 percentage of the protein. Then VLDL is mainly made up of the triacyl glycerol, and it has B100, C2, C1, and C3 apolipoproteins, and it has very high content of triacylglycerol, which is 90 to 93 percentage, and then the 7 to 10 percentage of the protein content. So in the same way, IDL has triacylglycerol and cholesterol, and LDL has cholesterol alone, and HDL has phospholipids as well as the cholesterol, sometimes cholesterol extra as well. And what are the signature apolipoproteins that you can get it from this whole table? So this is the overall structure that we can see in a nutshell. We can see that the density of the molecule will increase with this structure. So chylomicron has the very low density when compared with the HDL. And here they have given that how much triglyceride is there. For example, here it is around 98 percentage. Here it is 95. Here it is 90, and here it is very less, and here it is very less. So that is the difference between how different lipoproteins in the biological system are transferring or transporting the lipid molecules overall. So the chylomicrons are the low density that we have already told, and then the high density lipoproteins are the highest density. And this is the electrophoresis of the lipoprotein. And if you see that this is the cathode, and then this is the anode part, the high density will always have the more negatively charged amino acids. That's why it goes towards the anode. Whereas the chylomicron, which is having the less negatively charged molecule, that's why it is comes here. So chylomicron, LDL, and VLDL, and then the HDL are the electrophoretic pattern of lipoproteins. And plasma lipoproteins, we have already told there are different molecules will be transported that we have already seen it in the tabulation. Here I'm again telling you the chylomicron is responsible for the transport of triacylglycerol from the dietary sources, and whereas VLDL is responsible for the triacylglycerol of endogenous, for example, hepatic synthesis. And for cholesterol transport, we have two things. One is the LDL as well as the HDL, that low density lipoprotein as well as the high density lipoproteins. And important organs, if you see in the lipoprotein metabolisms, are the sources of lipoproteins itself, like intestine, it can be liver or extra hepatic tissues, and then the adipose tissue. The important enzymes, if you see the lipoprotein lipase, which form the free fatty acids from the triacylglycerol to glycerol and then free fatty acids. Then hepatic lipase will be there, and LCAT, which is the lecithin cholesterol acyltransferase. Yeah. So the next molecule is the lecithin cholesterol acyltransferase, which will form the lysolecithin while transferring the cholesterol molecule or the acyl molecule from the lysophospholipids, and CETP, which is called cholesterol ester transfer protein, which is also majorly transfers the ester molecules from the free fatty acids to cholesterol molecule to form the cholesterol esters, and apoproteins, and then the receptors majorly that we only talk about the LDL receptor in this case. First, we'll start with chylomicron metabolism, then we'll come to VLDL, then LDL, then finally HDL. These are very broad things, but I have made a concise idea about how each metabolism works in the lipoproteins. For example, we all know that the chylomicron usually assembled in the intestinal mucosa, and it has the lowest density and then the largest size in the overall lipoprotein molecules. As I already told, 99% of chylomicrons are lipids and only 1% is related to protein. And the characteristic upper protein of the chylomicron is B48. This is very important one mark in any exam. And usually see the chylomicrons are carry the dietary lipids from the intestine to the liver. And there are certain physiological milky appearance of the plasma will happen after the meal up to 2 hours because of this chylomicron transfer of tag molecules like triacylglycerol from the intestine to the liver through blood system. And this is the overall metabolism that we see. The first one is the dietary triacylglycerol will be taken up in the small intestine. From the small intestine, the TAG, the triacylglycerol, will be taken up by the nascent chylomicron. The nascent chylomicron will have the B48 apolipoprotein. And finally, from the HDL, it can take the ApoC and then the ApoE in this case. Then it will form the matured chylomicron. So this matured chylomicron, what it will do, it will go to the extra hepatic tissue where due to this ApoC and then the ApoE, it can get active. It can activate the lipoprotein lipase in the extrahepatic tissue. When the lipoprotein lipase is there, then the tag content, whatever the tag is there, the triacylglycerol, it will be converted to glycerol and then the free fatty acids. So this will be done by the lipoprotein lipase. Due to this activity, the reduced level of triacylglycerol will be there in the. Yeah. Due to this, reduced levels of triacylglycerol will be there in the system, and this forms the chylomicron remnant. This chylomicron remnant, this ApoA and then ApoC will go back to the HDL. This chylomicron remnant can be taken up into the liver by LRP receptor or this HL receptor. So finally, it can again form the cholesterol as well as the free fatty acids from this overall thing. So the lipoprotein lipase, if you see, actually I already told, which is located in the endothelial lineage of the blood vessels, and lipoprotein lipase is an extracellular enzyme and anchored by the heparin sulfate to capillary walls of the most tissues, and it is predominantly present in the adipose tissue, cardiac muscles, and then the skeletal muscle. This LPL required that I already told this ApoC is important for the lipoprotein lipase activity. This requires the ApoC2. So this ApoC2 will activate the lipoprotein lipase, whereas ApoC3 and then the ApoA2 inactivate the enzyme. This LPL degrades the tag into the glycerol as well as the free fatty acids by its activity. Whereas the insulin usually, because after the meal the insulin levels will go high, so that stimulates its synthesis and then the transfer to the luminal surface of the capillary. And overall, if you see, this is the overall mechanism, it happens. Then how this VLDL formed from here, that we are going to see. So the VLDL is responsible for the transfer of tag inside the endogenous form, whereas it's especially in the hepatic tissues. So it usually is biosynthesized in the hepatocytes as well as in the intestinal mucosal cells. And the signature for this VLDL, usually in the chylomechan is B48, but in the VLDL its cases it's a B100. And the VLDL predominantly contains triacylglycerol of the endogenous origin, and it facilitates the mobilizing out the endogenously synthesized lipids in the hepatocytes to the extrahepatic tissues. So this is the overall thing, the way how the chylomicrons transfers, how the it forms the nascent form, then it will form the mature form. The same way from the liver, it can take up the triacylglycerol as well as the C. It has the marker of B100. It's called nascent VLDL. This from the nascent VLDL, it can take up the ApoC and then the ApoE from the HDL. Then it form the matured VLDL. This matured VLDL, we already know that it contains the ApoC. ApoC can activate the lipoprotein lipase. Then it forms the free fatty acid as well as the glycerol. After the free fatty acid and the glycerol, there is the decrease in the triacylglycerol content here that form the VLDL remnant. Otherwise, it called as the IDL. IDL is nothing but intermittent density lipoproteins. From here, if you remove the apoprotein E, then it forms LDL, which only contains the cholesterol. This LDL, again, it can get into the tissues by the action of LDL receptors. Okay, how that done? That we are going to see. See, LDL is usually originates from the VLDL remnant, and it contains the endogenous cholesterol and then the phospholipids and very minimal amount of the tag. And here also the apoprotein B100 plays the signature role in the LDL formation. The transport of this thing is usually the endogenously biosynthesized cholesterol from liver to the peripheral hepatic tissues. Okay, so how it happens, that we are going to see. The most of the LDL is 70% is metabolized in the liver, and the remaining 30% is in the hepatic tissue. And as I already told, this LDL is uptake through the LDL receptors. How it is happening? This is the overall LDL receptor. LDL receptor has a ligand binding domain of 292 amino acid, and the EGF precursor homology approximately the 400 amino acid. Then the O linked sugar is around the 58 amino acids. Finally, the membrane spanning domain will be there, and the inside the cytoplasmic tail of the 50 amino acid will be there. Here we have to note only one domain, that is ligand binding domain. This ligand binding domain contains negatively charged amino acid. So this negatively charged amino acid makes interaction with the ApoB100 of LDL. Here ApoB100 will have positively charged amino acid. So whenever this LDL receptor is having any kind of the interaction between the ApoB100 as well as the LDL receptor, it can taken up by the action called endocytosis. So this is the C. If you see, the LDL receptor is also named as a ApoB100 and then ApoE receptors. It's present on the cell surface, and usually it's a clathrin coatal to take up the or the do the endocytosis. and it's abundant in liver and then adrenals and fibroblasts, smooth muscles, cells of the vascular endothelium, and then the lymphocytes. And then the large transmembrane glycoprotein is called around 115 kilodalton, and it has the five domains that I've already explained. And usually it helps in the LDL uptake in macrophages as well as in the other cellular systems. And this is the overall LDL uptake will take. I had to say already told ApoB100 will have the positively charged amino acid. It can taken up by the LDL receptor where it has the negatively charged amino acid. And it is a receptor mediated endocytosis. When the endocytosis occurs, then it forms the endosome. This endosome can fuse with the lysosome to form phagosome, phagolysosome. Then there the enzymes is responsible for the uh, lipid digestion will be there. Then finally, it can form the amino acids from the lipoproteins, fatty acids, then the cholesterol esters is form the cholesterol droplets, and it can finally get back to the endoplasmic reticulum for the other function. Then it again forms the LDL receptor and then the Golgi synthesis. So the overall thing is the ApoB100 induced identification will be there, then the LDL receptor formation, then this complexes of the LDL and then the receptor internalization by the receptor mediated endocytosis, then the endolysosome formation, and lysosomal lipase and proteases degrade the LDL into free fatty acids and amino acids. Then the cholesterol then incorporates into the cell membrane or is stored as a cholesterol esters. And upper B100 degrades to the amino acid. The receptors are again recycled. Next, we are going to see about the metabolism of HDL. HDL work is it's to transfer the cholesterol overall. So this usually synthesized in the liver as well as in the intestine. And the nascent HDL is called pre-beta HDL, that we will see it in the next train. Like what is going to happen. That is of the hepatic and then the intestinal 30% is origin. And the hepatic HDL usually it has the discoidal shape and it has the ApoA, ApoC2 and then the ApoE. And surface is has the phospholipids and then the free cholesterol. And in the intestinal HDL it has only the ApoA, and ApoC and ApoE are transferred from the hepatic HDL. And this is the overall HDL metabolism that we can see. And it has the pre-beta HDL and then the nascent HDL form and then HDL3 and then the HDL2 form. First, we'll see the pre-beta HDL that is formed from the hepatic tissue mostly, and it is having the upper protein A1, and it is having the pre-beta HDL, and it can take the phospholipids as well as the cholesterol from the tissues by the action of ABCA1. It's a ATP binding protein, ABCA1 actually. Yeah, ATP binding acid protein A1, which can transfer the cholesterol from the tissues to the pre-beta HDL. When the pre-beta HDL takes up the more cholesterol, it's formed the discoidal HDL called. It has the phospholipid as well as the cholesterol, and that can get used by the LCAT. We'll come back to this LCAT how it happens. And there is one more protein called SRB1, and SRB1 is the scavenger receptor B1, and what it does, it can take up the transfer the cholesterol from the tissues to this discoidal HDL. Finally it forms the final HDL called HDL3. And this LCAT usually what it will do, it will transfer the acyl groups, then it form the cholesterol esters here. Then finally, from this cholesterol esters, LCAT will be removed. If the LCAT is removed, then the HDL2 will be formed. This HDL2 again by the action of SRB1. It is a dual transporter. It can again take up this HDL2 into the liver, and it can form the in terms of bile acids and then the bile C. Okay, and how it is usually happens that we are going to see it in the next slide. The major enzyme that we have seen it here is lecithin cholesterol transferase. This lecithin cholesterol transfer is what it will do. It will transfer the cholesterol like free fatty acid molecule or the acyl molecule from the lecithin to the cholesterol. So from here one molecule it's transferred into here. It forms a cholesterol ester, and the lysophospholipid is formed here. So LCAT is associated with the HDL lipoprotein, and LCAT esters the cholesterol and add to the disc-shaped nascent HDL to make sure HDL spherical, that is called HDL3. And converts the cholesterol and then the phosphatidyl choline of chylomicron and VLDL remnant to the C. And here ApoA1, ApoA4, and ApoC1 stimulates the LCAT activity of the HDL. That's what we are seeing. Here the A1 is there, then it can give the so many LCAT activity. There is one more protein called the cholesterol ester transfer protein, which usually helps in the transfer between the tag from the VLDL to LDL to the HDL, and then the exchange of the cholesterol esters from the HDL to VLDL. That will be done by the CETP. So CETP activity usually reduce the content of the cholesterol ester of the HDL to form the HDL2, and transfers the HDL2 to the liver. And CETP activity is induced when there is need of the cholesterol to the extrahepatic tissue. If you see here from the HDL, cholesterol esters are transferred to the VLDL, but the triacylglycerols are transferred from the VLDL to here. The same LDL will share the triacylglycerol this side, and then the cholesterol esters this side by the cholesterol ester transfer protein. The same done by this between the LDL as well as the VLDL. And this is the overall form. From the pre-beta HDL, LCAT, what it will do, the cholesterol will be converted into the cholesterol esters, and by the action of CETP, it can take up the more triacylglycerol as well as the reduced cholesterol esters levels. That leads to the HDL2. This HDL2, by the action of SRB2, sorry, SRB1, it can take up to the uh, liver. So CETP is responsible for the formation of the subtraction of HDL, that is HDL2 and then the HDL3. And prior to the CTEP activity, HDL is smaller particle termed as HDL3, which is cholesterol ester rich. That's what I already told. Post CETP activity, HDL3 become the larger tag rich and termed as HDL2, and CETP, LCAT are interrelated. And this is the overall activity we see. From the HDL, it forms the ApoC2, ApoA, and then the ApoE. From the nascent chylomicron, and then the chylomicron, and then the VLDL, it can take up the triacylglycerol more. And by the action of LCAT activity, it can convert the cholesterol to the cholesterol esters, and then it can take up the VLDL from the VLDL or the LDL, whatever it is. It can take up the triacylglycerol, and then it transfer the CE here. And finally, it can take the HDL2 action. Then it will go back to the liver again. And this is the overall mechanism. If you see how the lipoprotein levels are activated in the system, and from the dietary lipids it forms the chylomicron. From the tissues or the liver, it can taken up by the VLDLs. Then how the chylomicron remnances are formed, and then ideal forms from the VLDL, and then the LDL forms from the ideal. Then the HDL can take up the cholesterol from the peripheral tissues to the liver. This is what the overall roadmap of cholesterol inside the body tissues. There is one more thing called reversal cholesterol transport, because if the cell is having more cholesterol, or the liver is not having the cholesterol in the body, or the dietary uptake of the cholesterol is not there in the body, then the reversal cholesterol transport should happen. Means it should take up the cholesterol from the tissues, which means the extrahepatic tissues, to the liver. So it can be done by the two proteins which we have already talked in the previous slide. One is the scavenger receptor binding protein called SRB1, and then the ATP binding cassette transport proteins. What it usually does, will start with SRB1. So SRB1, it's a, I already told, it's a bifunctional, or it's have the dual property. From the cells like the macrophage, it can transfer the cholesterol from here to the HDL2, and HDL2 or the HDL. From here, HDL, it can transfer the cholesterol again into the liver. This is the one mechanism that usually uh, occurs. The second mechanism is by the ABC transporters. Here, ABC1, usually what it does, it will take up the cholesterol molecule from the cells, or in the case of the liver, it can transfer it to the pre-beta HDL. And pre-beta HDL is again forms HDL2. And this is the main reason this ABCA1 is transfers the cholesterol to the pre-beta HDL, whereas APCG1, that usually transfers the cholesterol to the HDL3 or the discoidal HDL. And if it is forms the cholesterol or it transfer the cholesterol from the outside to the HDL, again it will go back to the liver. So overall, the reversal of the cholesterol transport is done by the two proteins. One is the SRB1, or else it can be the ABC transporter proteins. Whenever the cholesterol content is very low in the body, liver has to get the any kind of cholesterol molecule. That time, this reversal of the cholesterol transport takes place in the body. And then we will see the what are the different disorders related to this lipoprotein metabolism. This lipoproteinemia is nothing but it's a comparison of both hypo and then the hyper lipoproteinemias. And we'll first see the hypolipoproteinemias. In the first case is the A beta lipoproteinemia. A beta lipoproteinemia is that there is a defect in the loading of the ApoB in the content. So the ApoB is not there, means ApoB is important for the chylomicron formation, which is the B48, and VLDL and LDL needs the ApoB 100. If this is not there, then the chylomicron, those things are not formed. That leads to there are so many diseases related to the lipoproteins such as the blood acrylglycerol, and that glow will be there intestine, and then the liver accumulates the acylglycerols because this formation of the lipoprotein molecule will not be formed. And next is called familial alpha lipoprotein deficiency. There are three deficiencies that we usually talk, that is one is Tangier disease, another one is the fish eye disease, and then the ApoA1 deficiency. And Tangier diseases are related to the AB CA1 transporter deficiency, and fish eye disease is related to partial LCAT deficiency, and ApoA1 is related to the pre-beta HDL formation deficiency. So this all are related to the all are the low or the near absence of the HDL molecule. This is having the tendency towards the hypertriacylglycerolemia as a result of absence of the ApoC2, causing the inactive LPL will be takes place, lipoprotein, and then low LDL levels will be maintained, and atherosclerosis in the elderly places people have seen. The other diseases, if you see, hyperlipoproteinemias will happen, that related to the familial lipoprotein lipase deficiency, that causes the inactive LPL, and type 2A will be there, that leads to the defective LDL receptors or the mutation in the Licon region of the ApoB100. And third is the familial type 3 lipoproteinemia. Usually, if you see, this is deficiency in the remnant clearance by the liver due to the abnormality in the ApoE protein, and patients lack these isoforms of E3, and then the E4 have only the E2, which does not react with the E receptor. And we'll see that what are the remarks will happen. Increase in the chylomicron, the VLDL remnants of the density is less than 1.019 and causes the hypercholesterolemia, xanthomas, and then the atherosclerosis. And again, the type 4 will be that type 4 is a majorly it's related to the overproduction of the VLDL, and it's often associated with the glucose intolerance as well as the hyperinsulinemia. And then another one is a familial hyper alpha lipoproteinemia, which usually related to the increased concentration of HDL. If you see, there is no issue for in such case in this alpha lipoproteinemia that leads to the rare condition called beneficial to the health and the longevity. And if it is hepatic lipase, if it is not there, that deficiency leads to the accumulation of the large triglycerol rich HDL and then VLDL dominance. And these people have the extra xanthomas and then the coronary heart disease. And finally, the familial lecithin cholesterol acyl transferase deficiency. This absence of LCAT leads to the block in the reverse cholesterol transport, and then the HDL remains as an nascent disc incapable of the taking up in the estrogen, the cholesterol. And finally, familial lipoprotein excess, that is LPA, that we have never talked about in the overall, that consists of the 1 mole of LDL attached to the 1 mole of ApoA. ApoA shows the structural homologies to the plasminogen. If this, because of the more LPA, whenever any kind of uh, plaque kind of thing form, it cannot be cleared because of this LPA, because it acts as a plasminogen. Plasminogen is majorly will not or it facilitate the clot formation. So it mimics the plasminogen activity. So the premature coronary heart disease due to the atherosclerosis plus the thrombosis due to the inhibition of the fibrinolysis people have seen. So here we are going to see one such example for the atherosclerosis formation. Formation, how this lipid molecules or any kind of oxidative stress condition, it leads to the uh, 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 oxidized LDL formation, that is called oxidized LDL, that is low density lipoprotein. This oxidized low density lipoprotein, which reacts with the usually the LOX1 like protein, whenever the lipoprotein or else the LDL is more in the body, this kind of reaction takes place. What happens? This endothelial cells usually have the high surface expression of the lectin like oxidized low density lipoprotein. And what happens? This oxidized lipoprotein will binds to the LOX1 receptor. When it binds to the LOX1 receptor, the usual cases what will happen? It can increase or upregulate the scavengers like that. We have seen one scavenger, that is SRB1, but other scavengers like SRA1, CD36, and then the LOX1 will be upregulated. This leads to the late exosome and then the lysosome acid lipase degrade the cholesterol esters. And in the endoplasmic reticulum, this acyl coenzyme A, cholesterol acyl transferase, that contributes to the formation of the cholesterol esters from the free cholesterol. This cholesterol esters accumulates in the ER usually. What happens? This A cat also will be increased, and then the more cholesterol esters will be there. In the normal cases, this neutral cholesterol esters are hydrolyzed by the EH enzyme, and it's liberating the free cholesterol. But in this kind of oxidative stress condition, especially in the macrophage, more number of oxidated or oxidized LDL leads to the LOX1 upregulation and then the reduced levels of NCEH. Not only this, the enzymes related to the reverse cholesterol like ABCA1, ABCG, and SRB1 all are getting downregulated during this oxidative stress, as well as more oxidized LDL is present. And this leads to the more cholesterol esters in the ER that forms the foam cell of the macrophages. Especially here, we are talking about the macrophage. And this foam cell can form the or the thinning of the blood vessel that leads to the atherosclerotic plaques. So the foam cell become the trapped in the walls of the blood vessels and contribute to the formation of the atherosclerotic plaques and causes narrowing of the arteries, which can lead to the myocardial infarction or the heart attacks. The next example that we can see it in the lecture, you can go back and then check. And this is what the overall lipoprotein metabolism that we have checked. So next we'll come back to the beta oxidation of the fatty acids. First we'll see how the mobilization of the stored fat occurs. Then we can get into the beta oxidation, each and everything. What is the beta oxidation occurs? What are the enzymes responsible for the beta oxidation? Then how the odd chain fatty acids are metabolized in this case that we are going to see. Majorly, any neutral lipids are stored in the adipocytes and it's stored synthesized cells of the adrenal cortex. Ovary testis in the form of lipid droplets. It's none other than adiposome. This adiposome has the core structure of the cholesterol other than the neutral lipids, and it's surrounded by the monolayer of the phospholipids. Okay. Then, moreover, this lipids can further surrounded by the one of the in protein called perilipins. Okay, how the perilipins happens? This perilipins mobilization is usually from the hormones, epinephrines or the glucagon. So whenever this should be broken down and then it has to go for the glucosynthesis or the breakdown of the lipids, the hormone goes and binds to the receptor that we have seen this ATP or the cyclic AMP pathway already. Then the G5 gets activated. Then the adenyl cyclic, what it will happen? It will convert the ATP to the cyclic AMP. And cyclic AMP in turn activate the protein kinase A. This protein kinase can phosphorylate two systems. One is it can phosphorylate the perilipin. Once the perilipin is activated or it's phosphorylated, it can recruit the phosphorylated hormone sensitive lipase, which is also phosphorylated by the pKa. So when the phospholipase is active, inside the lipid droplets, whatever the tag is there, that is triacylglycerol, that will be broken down into the glycerol as well as the free fatty acids. This free fatty acids can get into the circulation by the addition of the albumin usually. Then it can get into the cells where it can get the beta oxidation. So once the free fatty acid transporter helps in the transfer of the fatty acids inside the myocytes, then the beta oxidation occurs. Then the citric acid cycle respiratory sync, then the a large amount of ATP and then the CO2 molecule are formed. Once the glycerol formation that we have seen it in the from the last couple of slides, what the glycerol will happen? First, glycerol will be converted to glycerol 3-phosphate by the action of glycerol kinase. Then this glycerol 3-phosphate dehydrogenase forms the glycerol 3-phosphate to the dihydroxyacetone phosphate. Then again the triose phosphate isomerase forms diglyceraldehyde 3-phosphate from the dihydroxyacetone phosphate that in turns get into the glycolysis. And we'll see the fatty acid now. The free fatty acid, whatever it's from, it has to go for the oxidation. So that is called different types of oxidation that we see. That is the alpha oxidation, beta, and then the omega oxidation. The beta oxidation in this case, like how we are usually saying, it's go like if you go back to the chemistry and then we see the carboxyl group, whatever it is there here, the first carbon attached to the carboxyl group, it's called the alpha carbon. The second carbon is a beta carbon, and then the furthest in the like final carbon is called the omega carbon. So based upon the oxidation where it occurs, it can formulate the alpha oxidation or in the beta oxidation or the omega oxidation overall. So the beta oxidation of the fatty acid usually undergoes the oxidative removal of the successive two carbon units called as the acetyl CoA, and starting from the carboxyl end of the fatty acid chain. The process is known as the beta oxidation because the oxidation and the splitting occurs as the beta carbon. First, any molecule it should get activated before into the getting into the cell. This activation of the fatty acid happens in the cytoplasm. The activated form of the fatty acid is called fatty acid CoA. And this activation done by the coenzyme A complex, which contains the pantothenic acid as well as the beta mercaptoethanolamine. So this is the overall structure of the coenzyme A. So beta mercaptoethanolamine, if you see, beta alanine will be there, pantoic acid will be there, then the ADP will be there. How it acts actually, it depends on the ATP because the enzyme responsible for this activation of the fatty acid is fatty acyl CoA synthetase. The synthase and synthetase are differs based upon the uh, usage of the ATP. When the synthetase usually uses the ATP, whereas synthase usually doesn't. Then the fatty acyl CoA synthesis uses the ATP. Then it transfers the uh, CoA group, then it forms the fatty acyl CoA in the two-step reaction. First, it forms the fatty acyl adenylate. Then the second, the fatty acyl CoA synthetase by the removal of the AMP, it forms the fatty acyl CoA. So here, what will happen? The ATP will be converted into AMP and the inorganic phosphate. The both AMP and then the inorganic phosphate or the pyrophosphate goes hydrolysis, then forms the large negative delta G, which is called free energy that helps in the activation of the fatty acids. So the based upon the chain size of the fatty acid, this fatty acid co-synthetase also differs. There are the different for the short chain, medium chain, as well as the long chain fatty acid. This usually present in the outer mitochondrial membrane. The small chain fatty acids may also be activated by the thiophorase enzyme using the succinyl CoA. And it has to be transferred because usually the enzymes responsible for the beta oxidation are present in the mitochondrial inner membrane as well as the mitochondrial matrix. So that usually transfers there should be a subtle system where the cytoplasmic activated fatty acid should be transferred into the inner mitochondrial membrane. That will be done by the carnitine. So carnitine is usually made up of lysine as well as the methionine, and this is chemically called as the beta hydroxy gamma trimethyl amino ammonium butyrate, and this acts as a transporter. The reason is whatever the activated acyl CoA is there, These things are not permeable inside the in a mitochondrial membrane. So this usually abundant in the muscle, and then whenever if you see the serum or the tissue, it levels varies during the growth and then the pregnancy, and during the aging process also it varies. Bones are particularly affected adversely whenever the carnitine is not there. It leads to the osteoporosis in the elderly subjects. So this is the overall the carnitine shuttle system. It has the four different process. First one is whenever the acyl CoA is activated in the cytosol by the action of the carnitine acyl transferase 1. It transfers the acyl CoA from the cytosol to the interspace membrane in the mitochondria. And from there, there is one more translocase transfer will be there. This translocase, which will transfer the acyl carnitine to the mitochondrial matrix. Once it transfers inside the matrix, in the matrix there is one more enzyme called carnitine acyl transferase 2, that is CAT2, that transfers the carnitine or the split of the carnitine as well as the acyl CoA separately. So this acyl CoA can undergo now beta oxidation, whereas carnitine, whatever that is formed, it has to go back to the interspace membrane. That also done by the same uh, uh, transporter called translocase transporter. So we'll see the beta oxidation now overall what happens. So inside the mitochondria, the acyl CoA follows the four enzyme catalyzed reaction called. First one is it has to dehydrogenated, then the hydrated, then again the dehydrogenated followed by the thiolytic splitting of the two carbon unit, that is called acetyl CoA. The cycle is repeated again and again, and an acetyl CoA molecule is split off each time till the last two or three carbon units are left. The last two carbon units are usually the acetyl CoA or the three carbon unit usually for the propionyl CoA. So let's see how this happens. So this is the overall reaction. The one such example that we are given the palmitoyl CoA. So the palmitoyl CoA, it has this structure. So wherever the beta oxidation occurs in the beta form. So the first acyl CoA dehydrogenase remove the hydrogen atoms from the beta carbon, and as well as the alpha carbon form the FADH2. And here it is formed the trans configuration. So the enol CoA hydrolyze will be there. It will hydrolyze the beta carbon here. Then it formed the L beta hydroxy acyl CoA. And then L beta, it's formed the trans delta 2 enol CoA previous step. The second step is the enol CoA hydratase that forms the beta hydroxy acyl CoA. This beta hydroxy acyl CoA acted upon by the beta hydroxy acyl CoA dehydrogenase, where NAD plus is converted to NADH, that will go into the electron transport chain, and this also goes into the electron transport chain. And finally, from the beta keto acyl CoA. This beta keto acyl CoA, again in the presence of the acyl CoA acetyl transferase, one quash will come, then the acetyl CoA will be formed, then the acyl CoA again formed. This acyl CoA again go back to the beta oxidation. So this is the overall step. From the FAD linked dehydrogenase form FADH2, and NAD plus dependent dehydrogenase form NADH2. And overall, if you see how the overall cycle will happen, from the 18 carbon or the 16 carbon of the palmitoyl CoA, it forms 8 molecules of acetyl CoA, and it undergoes the 7 cycle of beta oxidation overall. And the energetics of beta oxidation, if you see, overall 8 acetyl CoA will be formed, and each acetyl CoA will leads to the 10 ATP, so that form the 80 ATP in this case. And 7 FDH2, and it forms the 10.5 ATP, and 7 NADH, it forms the 17.5 ATP. Gross total is 108, whereas whenever the it get activated, there are 2 ATP molecules are utilized. So that's why 106 ATP. So if you see the palmitic acid, which undergoes the 7 cycles of beta oxidation, and it forms the 8 molecules of acetyl CoA. In each cycle, 1 molecule of FDADH2 and 1 molecule of NADH are produced. So this enter the mitochondria through the electron transport chain, and during the initial activation reaction, equivalents of the two high energy bonds are utilized. So that's why 108 minus 2, 106 ATP is happens. The efficiency of the beta oxidation is about 33% in the cell. So till now we have seen the even chain fatty acids. What happens if it is a odd chain fatty acids? Till the 5 carbon step, the last step, it continues the beta oxidation. Whereas the 5 carbon step, it will split into the 3 carbon as well as the 2 carbon acetyl CoA. This 3 carbon is nothing called propionyl CoA. This propionyl CoA, by the action of propionyl CoA carboxylase, in the presence of the biotin, it forms the dimethylmalonyl CoA. This methylmalonyl CoA epimerase, it forms the methylmalonyl CoA L. This L-methylmalonyl CoA, by the action of methylmalonyl CoA mutase, it forms the succinyl CoA, then it can enter the TCA cycle. So here the coenzyme B12 is really important for the methylmalonyl CoA mutase. If the deficiency, if the B12 is there, then the more accumulation of this propionyl CoA there in the cells that people have seen. And there is some more called unsaturated fatty acid. There are more unsaturated fatty acids are there in the body system. Unsaturated fatty acids are nothing but it has the double bond. So it is in the sixth configuration. Whenever the sixth configuration is there, it cannot be done because overall the enzymes that responds for the beta oxidation are trans configuration. So wherever the sixth configuration is there, then we have to use two more enzymes called auxiliary enzymes, that is called isomerase as well as the reductase. So here, if you see one of the unsaturated fatty acid is oleate, which is having the double bond at 9 and then 10th carbon. Okay, so till the ninth carbon, that means the three set of the beta oxidation will take place. And finally, forms a structure called 6-del-3 dodecynyl CoA. This dodecynyl CoA undergoes the isomerase reaction. Then this 6 configuration will be formed as a trans configuration. Then it comes back to the beta oxidation of the 5 cycles. Then the 6-acetyl CoA will be formed. There is a another enzyme called the reductase that forms whenever the more than two double bonds or the unsaturated fatty or the polyunsaturated fatty acids are there. So the regulation of the beta oxidation, if you see, the really important molecule in the overall beta oxidation step is carnitine transfer. Because carnitine transfer, if it is not there in the system, then will not be any kind of transport will not be there. Once the carnitine transport, if it is there, then the beta oxidation will definitely take place. But the high control of the carnitine acyl transfer is, or the carnitine molecule is really important. That done by the component called melanyl CoA. Melanyl CoA is formed during the fatty acid synthesis. So whenever the more fatty acid is synthesis happens, the melanyl CoA levels are more, then it inhibits the enzyme called carnitine acyl transferase, and thereby it can regulate the overall beta oxidation. There is other regulations called NADH and then the NAD plus ratio. If it is less, then the beta hydroxyl co-DH will be there. Then the acetyl CoA also it will reduce the beta oxidation. The AMP dependent regulation also it comes in the case of the short term regulation. When you see long-term regulation, there is a two receptor responsible for the long-term regulation. One is the peroxisome proliferator activated receptor family, and second one is the glucagon acts through the CAMP and then the CREB pathway. And we'll get back to the questions of the day. And what is the net ATP count from the complete oxidation of the one molecule of the palmitic acid? One molecule of palmitic acid, we know it forms 8 acetyl CoA, 7 NADH, and 7 FADH. 8 acetyl CoA form 10 ATP, which is called AT. 7 NADH 2.5. This is 1.5.
So net will
And
be
you,
106.
yeah, it's 1.6. Usually it forms 108, where in the activation step we lost 2, so it forms 116.
So if they ask total, it is 108 and the net is 106. No?
Yeah, net is 106.
No.
Net to be you know how we calculate net actually?
How is that? Minus 2, no?
Okay. Yeah, minus 2. Why are we doing minus 2 here?
For the initial activation.
Yeah, net is nothing but overall that pathway activated, how much we are having the less. For example,
Hmm.
say when you are joining any job, you are paying 1,000 rupees. But
Hmm.
when you are getting the salary, you are getting 10,000 rupees.
Hmm.
So usually you will get 9,000 rupees na, as a salary because 1,000 rupees you have already paid.
Yes, yes, yes.
So that is called net ATP count here.
Hmm.
Second one is carnitine formed from which two amino acids?
Lysine, methionine.
Yeah, lysine and methionine. And what is the marker for chylomicron?
B48.
Yeah, it's B48. B100 is the marker for
VLDL.
VLDL and
LDL.
LDL. Yeah, yeah. VLDL is remnant is called as
Hmm. That's ideal.
yeah ideal. Then final question. Which of the following factor responsible for lipid molecule transfer between lipoproteins? Which of the
Lipid.
following factor responsible?
Cholesterol. Hmm.
Ah,
LCAT also transfers the cholesterol. No,
but between
CETP.
lipoproteins.
Between that's CETP. CETP.
CETP. LCAT usually it converts the
Esteric.
cholesterol to cholesterol
Hmm.
esters
Hmm.
actually, not
Yes,
in the
yes.
transport reaction.
Yeah.
Yeah, that's what I have for the day. You can go back to the professor's lectures also if you have any doubt. Then
Okay. Yeah. Thank
do you
you.
have any doubt?
No, actually, the one question was there in the
Yeah,
questions which were asked. No, so in
yeah,
that one question was Tangier disease.
yeah. Actually, I also thought the same thing.
It's
I've
wrong.
gone
It's a,
through.
there's no option for that. It's wrong.
Yes, yes, yes. I will write it to the faculty actually, and you can
Yeah,
also
because
write it to the faculty. It's not an issue. I'll write it to the
hmm,
NPTL also. I will ask them to give the marks. For that.
they have to give because the option is not there.
Yes, yes, yes. I have checked it. I will write it to the professor then,
Yeah.
as well as the NPTL.
Okay. Yeah.
Yeah.
Thank you, sir.
Thank you so
Yeah.
much.
Bye.
We'll see you next week.
Okay. Bye.
And one more thing, from not next week, from the
Hmm.
next to next week, I'm shifting the classes to Saturday.
All
That
right.
I'll
All right.
inform it through the NPTL.
All right. All right.
Yeah, yeah.
No problem.
Thanks.
Thank you. Thank you.
Thank you.
Yeah, no mention.